Hi everyone, my name is Katja Kochvar, and today I will be presenting on my honors thesis research at Duke University on delayed maturation of song in a songbird. So a bit of background first about song in many songbirds. So usually there's this early life sensitive phase of learning. As you can see in this diagram, it goes from early discrimination to listening, practicing, and ultimately creating crystallized adult song. For this process to work well, usually conspecific adult song feedback is necessary. And then adult song can be used in both male-to-male competition and mate attraction contexts. So in both contexts, song is super important. And the main reason why is that it conveys useful information about the quality of the individual to receivers. And it's a reliable indicator because of the handicap principle as proposed by the developmental stress hypothesis. And this basically just posits that there is a relationship between the early life chick conditions that one experiences, the development of the brain, and thus the ability to produce quality song as an adult. As an adult, song can signal resource holding potential and/or aggressive intent in territory defense interactions between males, and it can serve a purpose in mate choice. So females that pay attention to male song can boost their reproductive success. So my study species was song sparrows. Just a couple facts about them. They are age-limited learners, so they have a critical sensitive period, and after that time, they don't really learn new song. They experience overproduction and selective attrition of song types, so they learn more songs during this critical period than they actually include in the repertoire as an adult. Their song is relatively complex. It consists of these alternating trills and note complexes, and we know that males and females respond to various parameters of song in various contexts. The main question we were interested in is whether delayed maturation of song occurs in this species. And delayed maturation is basically just age-related development corresponding to an increase in quality of a trait. And this has been established in many passerines, so we know that's the case in swamp sparrows, great reed warblers, and pied flatcatchers, among others. However, in song sparrows, only song repertoire has been examined, and there is actually no evidence of delayed maturation in either composition or size of song repertoire. So it's relatively unknown whether this occurs in song sparrow song. So we investigated this question using 17 birds hand-reared in the lab, and we had song recording data on the first 3 years of life of all of these birds. So it was a longitudinal data set. And we had recordings of at least 200 songs per bird per year and ended up analyzing over 50,000 songs. So this basically here is just a sample sonogram of a song sparrow song. And as you can see, like I mentioned before, there's these alternating note complexes and trills. And this is what we use to calculate all of our various measurements of song. So we ended up looking at 8 different song characteristics, 3 of which are measures of song complexity. So the first being within song type variation, which is basically calculated as the percentage of song variants of a type that are the most common variant, the average number of notes, and the number of unique note types, both of which were just for the most common variant. And then we had 5 different measures of song production patterns, which included singing rate, time interval between songs, both within a bout, so same song type, and between bouts, switching between song types, within song stereotypy, and between song consistency. And what we found were that 7 of those 8 characteristics significantly changed between ages 1 and 2, but none significantly changed between ages 2 and 3. And that's what you can see in this box plot here. So for the most common variant proportion, there was a significant change between ages 1 and 2. Same with number of notes, unique note types, within bout and between bout time interval, singing rate, and between song consistency. And the direction of that change changed. Sometimes it was in the predicted direction of increased quality, sometimes not, but either way, 7 of those 8 significantly changed between ages 1 and 2. The next thing we did was took all of those 8 characteristics and looked at whether a linear discriminant function analysis could distinguish between differently aged birds. And what we found was that it could easily distinguish between 1-year-old birds and what we will consider older birds, so 2- and 3-year-old birds lumped together. And there's a very distinct separation here. However, the model was not able to distinguish between 2-year-old and 3-year-old birds. So you can see in this density plot here, there is a significant overlap between 2-year-old and 3-year-old aged song. So the implications for this are quite straightforward. We found that song may be used as a reliable indicator of age, but really only distinguishing between young 1-year-old birds and 2- and 3-year-old birds, older birds as a group. And this is consistent with a peak in male quality at age 2 to 3, which is expected from experimental and theoretical evidence in song sparrows. So some future directions for this research. The first is definitely playback experiments, and this would help determine if male and/or female receivers can distinguish between differently aged song or have age-related song preferences. Also, age-related decreases in song quality would be interesting to look at. So senescence of song, which has been established in a few other species like swamp sparrows. And also comparing this work with wild song sparrow song development, because of course there are some differences between the hand rearing process and wild development of song. And looking at whether we can really replicate these results in the field would be of much interest. I would like to, of course, take the time to acknowledge my 3 future co-authors, my former PI, Steve Nowicki, Matthew Zippel, and Susan Peters. They are all wonderful mentors, human scientists, and I'm incredibly grateful to them. And with that, these are my references. Thank you so much, and I look forward to seeing you in the Q&A and live talk.